that many, but I hope it covers everything, like in the whole chapter. So the first question is from May, June 2021. The question states, much success was made in reducing the number of cases of malaria between 2000 and 2015. The question asks to explain how malaria is transmitted. So the question is worth 2 marks over here. And I would like to open this question to the floor. If any of you would like to try. If you know enough of this mosquito. Yes, that is correct. So first, you can talk about the vector. So over here, what you can say is the vector is Anopheles mosquito. And then the second point, what you can say is what does the Anopheles mosquito do? So what it does is it basically it takes blood from an infected person. Now the mark scheme would give this this point 1 mark, so it's sufficient for 2 marks. But if you guys aren't comfortable with this, you can add another one. You guys can say the mosquito injects an uninfected person with the pathogen Plasmodium. Yeah, so there's the 2 marks. Moving on, here, figure 5.2 shows 2 diagnostic test strips for malaria. So on the left, we can see that it is the negative one, and on the right, it is a positive one. So the question states, a sample of blood from a person suspected. Sorry, can I move my cat for a while? A sample of blood from a person suspected of having malaria is put into the well labeled S, and a buffer solution is put into the well labeled A. So you put the blood into here, and a buffer solution is put into here. So the buffer solution moves the blood towards the result window over here, and a line at position C indicates that the test is working correctly. So a line at position T indicates the positive result for malaria. So this test strip is actually really similar to RPK for COVID test. So if you guys have done the COVID test before, it's actually pretty convenient. So I gave you guys one already, one advantages, which you guys want to give the other 3. No? Past results. Accurate. Yes, you guys are correct. So first one, results are accurate. Yeah, results are accurate. Number 2, results are obtained quickly. And the third one, you can say it is sheet. So the mark scheme, they give 10, but over here you can just choose 3 because it is worth 3 marks. So moving on.
Hang on. Show us the rest of the 7, please.
Oh, okay. So I gave you guys the 3 already. So the next 7 is doesn't need highly trained medical professional, doesn't need specialized equipment, easily transported, can ensure appropriate treatment is given immediately, malaria is confirmed, reduces unnecessary use of anti-malarial drugs, a small area for, I don't know what S means here. Oh, a small area for S. This one. Yeah. Only a small sample of blood needed. And it also can identify different species of Plasmodium. So yeah. Moving on. From the February-March 2020, a person with HIV or AIDS has a weakened immune system. This is because HIV infects cells of the immune system, in particular T helper cells. The pathogen can remain inactive within host cells. In some people, the pathogen becomes active and causes the number of T helper cells to decrease. So ART, antiretroviral therapy, is used to treat people who are infected with HIV, and ART aims to keep the number of T helper cells at a healthy level. So the question asks to state the full name of pathogen known as HIV. So this one is very straightforward. I'll just answer. Immunodeficiency virus. Deficiency. I think this is how you spell it. Oh, my bad. Check. I'll check again now. How do you spell this thing?
Correct.
Correct, right?
Immune. There's an IMMU.
Immuno.
Remove the extra U. Immunodeficient. Add one C after the I.
Oh.
Deficient. Shen is C-I-E-N, I think. C-I-E-N.
C-I-E-N.
Deficiency. Yeah, very good. Thank you.
So the next question is, explain why it is important that ART maintains a healthy number of T helper cells. This question is related to chapter 11, but I thought I might as well just add it in because it's related. So the question here is worth 3 marks. So what you can say is you can give 2 points stating the effect of having a low number of T helper cells. So according to the mark scheme, what you can say is if the T helper cell count is low, so low T helper cells can cause an increased risk of developing an infectious disease. The second one is you can say low levels of, because you guys know that T helper cells secrete cytokine, and if you guys have very, very little low T helper cells, you guys will secrete less cytokines. Okay, and then to get the third mark, what you can say is you can explain the role of cytokines. So according to the mark scheme, cytokine It stimulates B lymphocytes to produce antibodies. So this is already sufficient for you guys to get 3 marks. As you guys can see, any tree from T helper cell number is low. So you guys can give 2 points from this as what I've just given. And the third one, you can say the role of cytokine. I went with the fourth one, stimulates B lymphocytes. That is already sufficient. And then I added to produce antibodies. But if you want to talk about, if you want to continue from here, you can say, so lower concentrations of antibody is produced. And here are the rest of the answers. It stimulates T killer cells, so fewer infected cells killed, more time for pathogens to reproduce, and fewer memory cells to fight future infection. So over here, I thought I just explain or recap. So over here, you can see the APC. Once the T helper cells, it binds to the APC, it undergoes mitosis, and then it differentiates into active T helper cells and T helper memory cells. So the active T helper cells is secrete cytokines to stimulate mitosis of B helper cells. So without cytokine, B helper cells cannot secrete antibody. So it is important that our T helper cell count is actually quite high or sufficient. So the B helper cells can undergo mitosis and differentiate into B lymphocyte memory cells and plasma cells. So the plasma cells is the one that secretes antibody molecules. Moving on. Figure 5.1 shows global estimates of the percentage of people living with HIV who received treatment with ART in each year from 2000 to 2015, and the number of people who died from HIV or AIDS in each year from 2000 to 2015. So the key here, the bar graph, is the percentage of people living with HIV who received treatment, and the line is the number of deaths from HIV and AIDS. So describe the trends shown in figure 5.1. So over here, the question is just worth 3 marks, and you have 2 keys. So what you can do is you can either explain two points for this one from the percentage of people living HIV, and then 1 point from number of deaths, or vice versa. But according to the mark scheme, they give two points from here and then 1 point from here. Okay, so over here, what you can see that it doesn't actually increase a lot from 2000 to 2002. You can see that it just increases just a little bit, and then it decreases just a little bit, but overall I don't think there's much of a difference, and then only it increases after that. So over here you can say there is a low rate of increase for people living with HIV who receive treatment and then you state the year between 2000 and 2000. Well, over here it says in the mark scheme you can give either 2000 and 2004 or 2000 and 2003, but I'll just say 2000 and 2003. And then the second point, after that, the percentage of people living with HIV increases, as you guys can see. So, so to get the last point, you can say that you can talk about the number of deaths from HIV. As you guys can see over here, it increases just until 2005, 2006, and then it decreases after that. So you can say increase in HIV related deaths from 2000 to 2004. And then it decreases. Okay. So over here, you can see that receiving treatment, maximum we can give 2. Percentage of people living with HIV receiving treatment increases. And then you can say the low rate of increase between 2000 and 2003 or 2004. And then you can talk about the deaths, increase in HIV AIDS related death to 2004 to 2005, and then it decreases. Or it accepts peak in 2004, 2005.
Hang on, I think the quoting the data is important for this kind of question. Every time you're asked to describe a graph or table, you see there are 2 marks in the mark scheme that gives student who give supporting data, like the percentage. So you need to read the percentage from the y-axis. Yeah.
Okay.
Yeah.
So over here, according to the mark scheme, between 2000 and 2003, over here on the y-axis, it's just 3% or 4%. And then in 2015, the peak, it's about 45%. So you guys can add that into your answers for support. And then in death, for data to support, it starts at 1.5 million over here. Approximately 1.5 lah. But, and then it peaks at 2 million. And then it decreases to around 1.2 million. Yeah. So over here, moving on. Penicillin belongs to a group of antibiotics known as beta-lactams, which all act in the same way on bacteria. So describe how penicillin kills non-resistant bacteria. So this one is related to antibiotics. So penicillin is a type of antibiotics, and we should know that antibiotic, it acts on the bacterial cell wall. So I'll explain this slide first before I move on to the answer. So over here, it inhibits the synthesis of new cell wall. Yeah, as you guys can see, a bacterial cell wall contains peptidoglycan, or it is made up of peptidoglycan. In a peptidoglycan monomer, a peptidoglycan is made up of 2 sugar subunits, and one of the subunit has a short polypeptide chain. As you guys can see over here, this one is the short polypeptide chain. You guys don't have to know the name of this, by the way. Anyways, so when both of these are joined together, They form a long chain polymer with cross links formed. This colorful thing is the cross links. As you guys can see, it forms cross links. So this causes the cell wall to not move or stretch, which preventing from cell burst. So in order for the bacteria to expand, water will enter the bacteria through the peptidoglycan, as you guys can see over here. Water will enter over here, so it causes the bacteria to expand. And what causes the holes to form over here is the enzyme autolysin. It continues to form holes on the cell wall for cell expansion during growth. So when the enzyme, it inhibits peptidase, as we know, the enzyme catalyzes the synthesis of crosslinks between peptidoglycan. Oh, I forgot to mention this. When it inhibits the synthesis of a new cell wall, it inhibits the glycoprotein peptidase. Now, the enzyme, it catalyzes the synthesis of crosslinks between peptidoglycan. So this crosslink is catalyzed by the enzyme peptidase. So once penicillin, it acts on the peptidase, the cell wall weakens because water enters the cell and it continues to expand and it becomes weaker without the crosslinks. And then water continues to enter the cell by osmosis, and then the cell wall cannot withstand the hydrolytic pressure, and then the cell bursts. So you guys got that. So moving on, penicillin belongs to group antibiotics, and then describe how penicillin kills non-resistant bacteria. So for the first point, what we can say is penicillin inhibits enzyme, as you can see, enzyme peptidase, and then it alters the shape of active site, the enzyme, but I personally wouldn't write this. I don't know, because it's just, I don't know, I just wouldn't. Anyways, and then the peptidoglycan chains cannot link up and it stops crosslinks from forming. And without the crosslinks, the cell wall becomes weaker. And then I would move on, and then I would just say the cell wall bursts. But over here, you can say Turgor of cell not resisted by cell wall, or idea of inability to withstand increased internal pressure, which causes the cell wall to burst. So moving on to the last question, explain why penicillin does not affect viruses. Now we know virus is not a cell. It has no cellular structure. For example, it has no cell wall. Unlike the bacteria, it has peptidoglycan, and it has no cell surface membrane. So it is a no target for antibiotics. And some viruses have no enzymes. So the second one is it is metabolically inactive, or it has no metabolism because it is non-living outside the host cell. So basically it just dies outside the host cell. And yeah, it uses host cell metabolism enzymes inside the host cell to synthesize new viruses. So conclusion, antibiotic only effective against bacteria and fungi, and antibiotic doesn't work on viral protein code. So over here is just worth 2 marks. Explain why penicillin doesn't affect viruses. Pretty easy, pretty straightforward. You guys can just say viruses have no peptidoglycan wall. Viruses have no cell structure. Viruses have no metabolism. And then you get your 2 marks. Okay, I'm done. Thank you guys. I'll pass this to, to Daniel.
With the structured questions, now moving on to MCQ questions, which I have done in the form of a quiz.